Assalamualaikum and a very good morning to all who is joining us this morning at breakfast UMHealth. So I'm Dr. Faissal. I'm the head of Department in Orthopaedic Surgery, and I would like to introduce our speaker for this morning, Dr. Tan Sit Loh. She's our senior lecturer and she's a scientist and also the program coordinator for our Masters of Medical Science in Regenerative Medicine. So I think she's going to give us a talk on mesenchymal stem cells in tendon regeneration. So I think without further ado, I think we're going to let her to start her presentations.
Prof. Assoc for the kind introduction. Share screen. Okay, so good morning, everyone. So today I will talk to you about mesenchymal stem cells in tendon regenerations, some of the ongoing work and some of the work that we have conducted in our research team. So first of all, let me have a brief introduction about what is a tendon. So tendon is a connective tissue that connected muscle to bone and allow our joints to move in the various direction. And this figure on this slide here is showing the hierarchical structure of the tendon tissue from the smallest collagen molecule, which bundles into the macrofibril, subfibril, fibril, and the fascicles and the several fascicles which bundles together to form the tendon tissues. And the table below here is showing some important proteins that is important in tendon. For example, the sclerosis, tenomodulin, and tendency C are the important key biomarkers to show a tenogenic characteristic. And type 1 collagen is the most abundant collagen type that is present in the tendon tissues. So the current tendon repair methods is arthroscopic repair with suture, and with this it has several limitations, which include the calf limitations and calf mobility, the size, stiffness of the tear area, and also the adequacy for the visualization during the procedure. And there is high recurrence rate post repair, and also the presence of the scarring tissue at the edge of the tendon may prevent proper tendon repair. And also the tendon usually do not heal to bone. Specifically for the osteotendinous junction healing, where fibrocartilage tissue usually observed. And what is the mesenchymal stem cell? So it is the cell that can self-renew and can able to differentiate into multiple lineages. And in this work that we are focusing or more interested in the tenogenic differentiation, which is the differentiation of the mesenchymal stem cells into the tenogenic lineage into the tendon cells or tenocytes. So this is the position paper which is published by the International Society for Cellular Therapy, which summarized the minimal criteria for defining the mesenchymal stem cells. So, and this has been, and since then, this has been used as the set of criteria to characterize the mesenchymal stem cell. And there are the plastic adherence characteristic and also the positive or negative expressions of the several CD markers as listed here, as well as the capability of the cells to differentiate into the tri-lineages, including the osteocyte, adipocyte, and chondrocyte lineage. So with that, we first asked whether the GDF5, this growth factor, was able to be a tenogenic differentiation inducer to differentiate the mesenchymal stem cell to the tendon cells. So based on before we have started this in vitro work to induce the mesenchymal stem cell for tenogenic differentiations with the GDF5, some of the previous work that has published at that time was word expressions of the sclerosis gene in the mesenchymal stem cells has shown that the cell able to express the tenogenic expression profile, which include the upregulated type 1 collagen expression as well as the tenosyne C expressions. And with that, there's also another group which at that time has published with the effect of the 10% cyclic stretching on the mesenchymal stem cell, which show upregulated phenotypic expressions with the formations of the stress fibers, as shown in the actin stain here in this figure. So to investigate the effect of GDF5 on MSC tenogenic differentiations, we evaluate several tenogenic markers and the cytoskeleton reorganizations. So in this work, we isolated the mesenchymal stem cells from the bone marrow, and we characterized based on the criteria as defined by the ISCT criteria. And the tendon cells isolated from isolated from the hamstring tendon were used as a control in comparison to the other in vitro study group, which include the untreated MSC treated with GDF5, day 4 treated cells, and the day 5 treated cells. And the cells were harvested and analyzed with the gene expression profile analysis using the contingent platform, as well as confocal and FM imaging approach. So in this study, the cells were seated and viewed under high spatial resolution using this closed cell incubation sample plate. And with this closed cell incubation sample plate, the cells are visualized close to physiological conditions, with the cell incubated at 37 degrees Celsius and with the CO2 control in this small chamber that you can see here. And this chamber is mounted on a stage for the AFM imaging. And with that, we were able to visualize the formation of the stress fiber in the mesenchymal stem cell upon the stimulation by the growth factors. So from the contingent expression profile, we have found that the tenogenic markers were upregulated in the GDF5 induced MSC, and non-tenogenic markers such as chronogenic marker and osteogenic markers were downregulated. And these are the confocal images for the different tenogenic phenotypic markers. And confocal imaging shows the actin cytoskeleton reorganization in the MSC upon GDF5 induction, as you can see in the diagram here, the stress fiber formation pointed by the white arrow here. And in the high resolution AFM topography imaging, the GDF5 also present with the observed with the formations of the stress fiber, as indicated by the white arrow here. And this observation shadowed the observation that we seen in the confocal imaging. So with that, in summary, the GDF5 induced So in summary, GDF5 induced the reorganization of actin structure, which involved the F-actin polymerization and restructuring of the cortical actin elements to newly formed stress fiber in the MSC. And stress fiber formations is an important event in the tenogenesis, especially with GDF5 induced MSC. So this growth factor is able to alter the MSC tenogenic marker gene expressions, as well as the cytoskeleton reorganization, which direct the MSC into the tenocyte-like cells. And the relevance of these tenocyte in tendon tissue is that in the focal adhesions, the stress fiber which attach to the plasma membrane, the integrin, which is present on the plasma membrane, the extracellular domain of the integrin will link to the matrix protein in the ECM, which allows the tendon to attach firmly to these high tensional loading environment, the tendon tissues. Okay, with this, we would like to know whether the tendinogenic MSC will be a potential cell source for tendon repair. So, and these figures here summarize the different phase of tendon healing. So, as you know, there's different phase of healing, which include the inflammatory phase, proliferation phase, remodeling phase, as well as the maturation phase. Usually, the growth factors or stem cell or biomaterials will apply the healing at the proliferation stage. So in this part of work, that we investigate the effect of different cell-based therapies to include the tendon cells, MSC and tendinogenic MSC in the data curve tendon repair using a rapid in vivo model. So in this part of study, the part one is the isolation of and expansion of the MSC and the tendon cells. And part two is experiment where the tendon defect was created, and the tendon defect was created, and the defect was repaired with other with the different methods, as shown. With different survey approach. I will explain more in the following slide. So the isolated MSC were characterized based on the ISCT criteria, and it shows the cells isolated. But the MSC, and we induced the MSC into the tendinogenic lineage using the GDF5. And so in the preclinical in vivo study, a window defect was created in the infraspinatus tendon, and a silicone sheet was used to maintain the defect. And after 3 weeks, the defect was repaired with different approach, as shown in this, as summarized in the table here. Suture is a conventional repair technique. So we have the group that repair with the suture, and suture plus the fibrin glue, because the fibrin glue is that is used in the cell treated groups. So in the cell repair with the sutures with fibrils, either tendon cell or MSC treated with GDF5. So at the end point of the in vivo study, we observe tendon callus formations in the repair tendon and in the MSC, tendinogenic MSC treated group only. So in this, so the tendon callus formation indicates the tendon new formations in this DMSC treated group. And from the histology, microscopic observations, this on the left here is a histology representative for the different the tendon treated with a different treatment. And in as you can see here, the no repair control, you can see the tissue still presented with the random fibers or orientation, and it also show very high abundance of cellularity compared to those treated with the tendinocyte or MSC or the tendinogenic MSC. And in terms of the fiber orientation, as you can see in the normal tendon, it shows a parallel collagen fibers in the parallel orientations to the tendon's long axis. As compared to the MSC and tendinogenic MSC, you can see that it shows close to the parallel, very close to the native tendon tissues, parallels of collagen fibers orientations in the MSC treated and tendinogenic MSC treated group as compared to the control. And this indicates more mature tendon healing in this cell-based treated group compared to the no repair or compared to the suture treated group. And the right side here shows the scoring for the tendon repair based on the Watkin score and the Solowski score. For the Watkin score, the higher the score, the better the tendon healing outcome. In the Solowski score, the the lower the score, the better the healing outcome. And as in the previous slide, it mentioned about the summary of these different phases of tendon healing. And in this in vivo study, the endpoint of the in vivo study is at 6 weeks of the post repair. So at 6 weeks is in the tendon healing is at this remodeling stage, and from the histology observation that we observe from the cell-based treated group, we can see they presented with the close to mature tendon healing stage. So this is a table that summarizes this p-value score for the scoring for tendon healing. And from this scoring, there's no significant difference between these cell-based treated groups. So we further analyze this cell-based treated group using additional analysis where we include the gene expression analysis for the tissues from these groups, from the cell-based treated group compared to the control, and it shows significant upregulations of the type 1 collagen and sclerosis genes in the Tendon Regeneration treated group. And confocal imaging also shows enhanced or increased expression of type 1 collagen, sclerosis, tendency C, and tendon D in the cell-based treated group. FM imagings of the collagen fibers in the repair tendon show the collagen fibers that present in the repair tendon with more parallel orientations for the cell-based treated tendon group. So in summary, the TMSC treated tendons have superior tendon healing compared to tenocytes and MSC treated tendons. So the limitations of this current work that we have is the small sample size, and we have not, and because of the small sample size, we have not able to include the biomechanical testing to evaluate the function, the regain of functions of the tendon post repair. So future studies shall include some biomedical testing for different cell-based treated tendon, and in order to elucidate the return of or regain of function in the tendon following repair. So with the MSC or TMSC BL solutions for tendon adhesion program. So so one of the common tendon healing complications is adhesions and also the rupture. So with that, we further the study and look into the efficacy of the MSC seeded biomaterial, and in this case here we use the human immune as a biomaterial, and the MSC seeded hem is used to treat the tendon in the Achilles tendon model in the SD rats. In this study, the left tendon is no surgery was performed on the left Achilles tendon, and the right tendon created with a defect and either treated with the conventional suture or suture with plus the hem or human immune, and or suture plus the immune that is seeded BMSC. So this is some diagram for the surgery. And from the microscopic adhesions evaluations based on the tongue scores for the adhesions to assess whether the adhesions occurs in the repair tendon, and there is a significant reduce in the adhesions score in the immune or MSC seeded immune treated group at 6 weeks and at 9 weeks post repair. And microscopic tendon healing evaluations based on the solidity score is also show improved tendon healing in the MSC in the tendon treated with hem and tendon treated with the MSC seeded hem. So here is the summary of the histology from the different groups, and basically from the big nine observations, you can see that the tendon show more parallel orientations. The correction is a parallel orientation in the MSC seated hem treated group compared to the suture group or the group that's treated with the hem. So from the histology observation, also analyze for the cellularity in the repair tendon, and it shows a significant reduce in the cellularity in the MSC seated hem treated. Okay, so and from the cellularity analysis, we also observe a reduce in a significant reduce in the number of rounded cells, and these rounded cells are basically it's not the mature tendon cells. So so it shows a significant reduce of these rounded cells in the group treated with the MSC seated hem. And from the collagen content analysis, there is no significant difference. The time point that we in this study, so probably extended time frame allow us to observe more changes in the total collagen content in the tendon treated. And from the biomechanical testing, however, we observe no significant difference in the Young's modulus among the three treatment groups at 3 weeks, 6 weeks, and 9 weeks post repair. So hem and hem CWMSC allows less adhesions and more mobility in the repair tendon, and they also show better tendon healing and more collagen matrix and tendon fibers that is oriented tangentially or in the parallel orientation to the tendon's long axis, and more tendon cell maturations or higher number of elongated cells were observed in the repair tendon, the tendon that is treated with the hem or MSC seated hem. Moving on, we would like to discuss a little bit on some of the work in tendon repair related to tendon repair, some of the work related to diabetes and treatment with some of the biologics. So as we are aware of, there are in recent reports that about Malaysia is one of the fattest nation among Southeast Asia countries, and the prevalence of the type 2 diabetes in Asian countries is also Malaysia is also among the highest countries. So in the type 2 diabetes conditions, it increased the risk of tendon rupture and several heart disease through these conditions is the presence of the angiopathy and also immobility in the diabetes conditions. So with that, we'd like to look into the effect of the MSC-derived extracellular vehicles in the diabetics therapy. So in literature review, we have already known that the MSC EVs carries various bioactive molecules that would exert effective role in treatment of diabetic complications through multiple pathways. Various reports has been reported in the literature for the treatment of diabetic complications using the MSC EVs. So with that, we would like to investigate to determine the effects of MSC exosomes in modulating the type 2 diabetes associated tendon degenerations in SEZ-induced type 2 diabetes rat model. So in this study, the type 2 diabetes conditions were reduced in the SE rats, and the rats that diabetic rats either without treatment or is treated with the different dose of the MSC EV via intravenous delivery. So actually, this is a part of the study that we collaborate with another research team, which actually the study is looking into the effect of the intravenous delivery of MSC EV in diabetes conditions, how it enhance the diabetes conditions. And for ours work, we only focus on the effect of this EV in the degenerations in the diabetes model. So the tendons were collected at the endpoint and analyzed with histology analysis, total collagen assay, as well as the biomechanical testing. So from this work, based on the histology analysis, there's a significant reductions in the total cellularity in the tendon that is treated with MSC EVs. And for total collagen content, it shows an increase in the total collagen content in the MSC EV treated group in the diabetes tendon compared to the untreated tendon in the diabetes group. However, in the biomechanical testing, there is no significant difference in the maximum load and Young's modulus in the Achilles tendon with or without MSC EV treatment in the diabetes group. So in summary, MSC type 2 diabetes rat model show increase in the collagen fiber crimping and also rounded increase in the rounded tendinoblast-like cells in the type 2 diabetes tendon, and there's a significant decrease in the total cerebral collagen content in the type 2 diabetes tendon. And MSC exosomes or MSC EV treated groups show a significant reduce in the cellularity compared to the untreated group, and no significant difference in the biomechanical properties in the type 2 diabetes tendon at 3 weeks post type 2 diabetes inductions. And so future studies shall look into a larger sample size and extend the study to a longer durations. So with that, the summary of my whole presentation today. So GDF5 induced MSC are able to induce tendinogenic differentiation of MSC, and GDF5 induced MSC can promote or enhance better tendon healing. And with MSC seated hem, it can reduce the tendon adhesions and allows more mobility and also better tendon healing. And MSC exosomes or MSC EV reduce the cellularity in tendon in type 2 diabetes tendon. So with that, that's on my presentation. Thank you for your attention.
Okay, thank you so much, Dr. Tan Sik Lu. I think I've not seen any questions in the Q&A box. Just a quick questions based on your study and your research that you have done so far. Would you actually recommend these treatments for tendon repair to the surgeons?
So I would say, if for clinical application, I would say that the cell-based therapy is still at the research stage, from moving on from the preclinical model into the clinical trial. So to make it possible for the clinical use of this, we still need to move on further to the clinical trial. Then from that we can more confidently to know whether we can suggest this as a treatment for tendon repair in future.
Yeah, I think it looks promising treatments for tendon repair. Hopefully, with more findings in the clinical trials later on, so we can hopefully come up with some products that we can just inject into the tendon and then they get the repair without the surgery. So any side effect, Prof. Tengkusar was asking if you're going to treat it with the MSC, because I think sometimes they concern whether the MSC will actually differentiate to other cells rather than a tendon, right?
Yeah, very good questions. Thank you, Prof. Sarah, for the questions. Actually, that is one of the concern at the early stage when people start looking at cell-based therapy for MSC treatment in the tendon repair. That is one of the concern about whether the MSC will differentiate into other lineage, for example, ectopic bone formations that has been observed previously in the MSC treated UMHealth study. And so with that, that's the reason our study maybe started, we focus into to induce the MSC, to first induce the MSC into the tenogenic lineage before we use it for the tendon repair, so that the cell has been preconditions to differentiate into the tenogenic lineage, and so that it would not go into other lineages, because we know that the mesenchymal stem cell can differentiate into the different lineages. So with the tenogenic MSC, it would more into the tenogenic phenotype, and we use it for the tendon repair. And of course, so for other side effects, so it's also part of the required list to be tested moving on from preclinical to the clinical trial, is that to show the cells are safe and would not they would not go into any tumor formations, to show the safety before we can really use it for the clinical treatment for tendon repair.
Okay. Thank you so much. Because of the time limitation, there's several questions in the Q&A. Maybe Dr. Tan can answer that in the chat box. So I think we need to move on to the next sessions. Thank you so much for listening.